Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International GCSE Biology Paper 2B for June 2024. Let's begin with the first question. During the evolution of living organisms, most species have become extinct. Evolution by natural selection means that species constantly replace each other. The photograph shows the fossilized jaws of a shark called Megalodon. Megalodon was a giant predatory shark that became extinct 2.6 million years ago. Some scientists think Megalodon was replaced by giant predatory toothed whales. These whales were the ancestors of modern orcas. Any sharks that remained evolved to become smaller. As well as the steady loss of species over time, there have been mass extinction events. Approximately 250 million years ago, 90% of all animals and plants became extinct. The cause of this mass extinction is not known, but one theory is that it was due to the eruptions of volcanoes. The volcanoes released sulfur dioxide, carbon dioxide, and dust into the atmosphere. This caused populations of producer species to fall, atmospheric carbon dioxide levels to rise, and oxygen levels to fall. Human activity is now causing another mass extinction. Some scientists estimate that approximately 1 million animal and plant species are at risk. To prevent species loss, cryozoos are being developed. Cryozoos are storage tanks containing samples of body cells from animals and are kept at a temperature of -170 degrees Celsius. The cells are put in a salt and sugar solution and then frozen. If a species becomes extinct, the frozen cells can be used to clone new animals. The banting is an endangered breed of cattle. Banting body cells have been stored in a cryozoo. These cells have been used with eggs from another species to successfully produce a living banting. In the future, we may be able to bring back extinct species, such as mammoths, by making clones using the remains of frozen mammoths. Scientists need to consider the advantages and disadvantages of bringing back extinct animals. Currently, the biodiversity of the world is different from the time when mammoths were alive. Many natural predators of mammoths are now extinct. Mammoths may have helped to keep methane-releasing soil frozen, reducing the release of the methane. Mammoths were also an important part of the nitrogen cycle and increased the nutrient content of low-nutrient Arctic soil. So in part A they say, Megalodon was an animal. Which of these features does Megalodon share with a fungus yeast? Nuclei present in cells, that's correct. Nervous coordination, no they don't, and stores carbohydrate as glycogen, that is correct. So the answer is 1 and 3 only. In part B, scientists think that the giant predatory toothed whales that replaced the extinct Megalodon consumed the same sources of food. Explain how the evolution of giant predatory toothed whales may have caused the extinction of Megalodon. If they were having the same food resources and those that survived were able to reproduce and have more of their kind. So I say the giant predatory toothed whales likely competed with Megalodon for the same food sources. If the whales were more efficient hunters or adapted better to the environment, they ate more food and outcompeted the Megalodon. As a result, they survived and reproduced, having more offsprings while the Megalodons died out. And next, volcanoes release sulfur dioxide into the atmosphere. Describe the biological consequence of sulfur dioxide release. Sulfur dioxide can react with the water in the air to form sulfurous acid. It could also react with oxygen to form sulfur trioxide, which can later react with water when it rains to form sulfuric acid. So it leads to the formation of acid rain, and this acid rain can change the pH of the soil, the pH of the water, affecting the microorganisms that are decomposers, as well as changing the conditions for other organisms to survive properly. So I said sulfur dioxide released in the atmosphere can lead to the formation of acid rain. Acid rain can damage plants, especially their leaves, which reduces photosynthesis. It can also acidify water bodies, killing aquatic organisms and disrupting ecosystems. These effects harm producers, which are the base of the food chain, ultimately causing the collapse of biodiversity. Then next, the release of large amounts of dust into the atmosphere reduces light intensity. Explain why this would cause a loss of food chains. Light intensity is very important in photosynthesis. If the light intensity is decreased, then the rate of photosynthesis is going to go down and the producers are going to make less food. Then the consumers that depend on the producers are going to be affected because there'll be less food for them to eat. So I say dust particles block sunlight. This decrease in light reduces the rate of photosynthesis in plants and phytoplanktons, which are primary producers, or which are producers, you could say just producers. With fewer producers, herbivores have less food, leading to a decline in their population. This effect causes collapse in various food chains due to lack of energy flow to the consumers. Then in part D, explain why the cells in cryozoos are put in salt and sugar solution rather than pure water. If placed in pure water, then the cells are going to gain water by osmosis because the cytoplasm of the cells are going to have a higher solid concentration, and so the water will move in. So I say pure water would cause the cells to swell and burst due to osmosis, as water would enter the cells where solid concentration is higher. Salt and sugar solutions are isotonic or slightly hypertonic, preventing excessive water movement in or out of the cell, and this helps maintain the cell structure during freezing and storage. Describe how scientists could clone a banteng. To clone the banteng, you have to get a body cell of a banteng, get the nucleus and transfer it into an enucleated ovum, and then allow the ovum to go through mitosis, produce an embryo, and then the embryo is implanted into a surrogate animal. So I say scientists take the nucleus from a body cell of the banteng and insert it into an enucleated egg cell of a related species. This egg is then stimulated by electric shock to begin dividing and develop into an embryo. The embryo is then implanted into the uterus of a surrogate animal, where it develops into a cloned banteng. Then explain why releasing cloned mammoths could have negative and positive effects on Arctic biodiversity. We read that from line 22 to 28. The positive effects could be that mammoths could help maintain the Arctic ecosystems by compacting snow and keeping the soil frozen, which prevents methane release, hence a decrease in global warming. They may also enrich soil nutrients by promoting the nitrogen cycle, supporting plant growth and increasing biodiversity, because the nitrogen cycle leads to formation of more proteins, of course more amino acids, more proteins, more DNA, more RNA, and so on. Then the negative effects, the current Arctic environment has changed since mammoths last lived, so their reintroduction could disrupt modern ecosystems. They might compete with existing species for food and territory, altering the balance of the ecosystem as well. Then in question 2, transpiration, which is water loss, is affected by several environmental factors. A student investigates the effect of light intensity on water loss from a plant shoot. The diagram shows some of the apparatus the student uses. So this is the setup. We see the lamp to provide the light, and then water is going to go up through the chute during transpiration. The student predicts that water loss from a plant shoot will increase as light intensity increases. Describe how to set up and use the student's apparatus to test this prediction. So you set up the potometer, which is the apparatus as shown in the diagram, ensuring it's airtight to prevent leaks. You cut and attach the healthy plant shoot to the apparatus underwater to avoid air bubbles. Place the apparatus near a lamp to provide light and measure the distance between the lamp and the plant to control light intensity. This is because you're going to move the lamp away as a method of varying light intensity. And then you mark the initial position of the air bubble in the capillary tube. After a set of time, this could be 10 minutes, measure how far the bubble has moved. This shows how much water was taken up. Of course, if you know the diameter of this tube and you know the length, you can be able to calculate the volume of water that has been taken up. And then repeat the experiment at different distances or using different wattage bulbs to change light intensity. You keep other variables like temperature, wind speed, and humidity constant. Set the dependent variable in this investigation. It could be the mean volume of water taken up by the chute or distance moved by the air bubble in a certain time. In part B, another student uses the apparatus to collect and process the data. The table shows their results. We see as light intensity increases, the mean volume of water lost through the chute increases in the 10 minutes, but after this light intensity, we see it does not change at all. So this explains why the light intensity changes the mean volume of water lost by the chute. Simple, as light intensity increases, the stomata open up more, and so more water is going to be lost through transpiration. So I said as light intensity increases, the stomata open wider to allow more carbon dioxide in for photosynthesis. This also increases transpiration. As more water evaporates from the leaf surface until all the stomata are open, then water loss is going to be constant, like at this point here. Then the student repeats the experiment with a plant that is adapted to live in a desert environment. So the table 2 shows the results. We see as light intensity increases, there is a decrease in the water loss, and after this light intensity, there is no change or there is no water loss at all. So they say the desert plant has adaptations to survive in desert environments where not much water is available. Explain why the results for this desert plant in table 2 are different from those in table 1. This could be because this plant is adapted to close the stomata in order to decrease water loss as light intensity increases, preventing excessive water loss during the desert heat. So I said the desert plant is adapted to conserve water in dry conditions. It may have fewer stomata or sunken stomata or thick waxy cuticle, all of which reduce water loss. Some desert plants may close their stomata during the day to avoid losing water in high heat. As a result, even at high light intensity, they lose less water or no water, unlike the plant in table 1, which lacks such adaptations. In question 3, isotonic drinks are often used to rehydrate athletes after exercise. Explain why athletes lose water during exercise. This could be due to sweating. As they exercise, the body temperature increases, and then water is going to be lost as a way of regulating the body temperature. So I say during exercise, the body temperature increases due to increased metabolism. Athletes lose water through sweating, which helps cool the body down through evaporation. And in part B, isotonic drinks contain salt and sugar solutions that are the same concentration as normal blood plasma. A scientist uses this method to investigate how well an isotonic drink rehydrates athletes. 3 athletes exercise for 1 hour. One athlete has no drink. The second athlete drinks 1,000 centimeters cubed of pure water. The third athlete drinks 1,000 centimeters cubed of an isotonic drink. The volume and color of urine produced by each athlete are measured 1 hour later. So from the table of results, we see with no drink, the volume of urine produced was 100 centimeters cubed, and we see it's very dark yellow. If you compare with the others, this is going to be the least. It's because they had no drink, and yet they were sweating during exercise, so their body is trying to conserve more water. ADH is going to be released, more water is going to be reabsorbed, and so the concentration of their urine is going to increase while the volume is going to go down. With pure water, they are more hydrated. Even if they do sweat, ADH is not going to be released as much, so less conservation or less reabsorption of water occurs. So the urine they produce is going to be very light yellow. It's going to be at a lower concentration and larger volume. That's why we see these results. And for the isotonic drink, this is similar to the concentration of plasma in terms of ion concentration. So we see the volume of urine produced is going to be 500 centimeters cubed, and the color of urine is going to be yellow. Because the isotonic drink has some ions in it, it will slightly increase the concentration or the amount of ions within the body. So some of those have to be released out. That's why we see the volume of urine is kind of higher than that, because they took in some water and more ions. And so the color is not as yellow as that, meaning it's a little concentrated than that, but not as concentrated as the first one. Then they say explain the results shown in the table for the athlete who consumed no drink. So I say that osmoreceptors in the hypothalamus detected the increase in solid concentration of the blood as water loss through sweating occurred. The pituitary gland was stimulated to release more ADH. ADH increased the permeability of the collecting ducts, leading to reabsorption of water. So the concentration of urine increased and the volume decreased, leading to the very dark yellow color. Then explain the differences in urine volume and urine color produced by the athlete who consumed the isotonic drink and the athlete who consumed pure water. I've already explained the results in the table. The athlete who drank pure water produced more urine because water is hypotonic and gets excreted quickly to maintain the balance of salts in the body. No ADH was released, so less water was reabsorbed by the collecting ducts, and the urine was very light yellow in color. In question 4, the diagram shows 4 glands in the human body labeled W, X, Y, and Z. So they say LH is a hormone involved in the menstrual cycle. Which labeled gland produces LH? It's the pituitary gland. So the answer should be W, which is that gland there. Then they say describe the function of LH during the menstrual cycle. LH triggers ovulation and it stimulates the production of progesterone by the ovaries, which maintains the uterine lining during the cycle. Then in part B, the diagram shows a fetus developing in a uterus. This is the diagram they have. Explain how the amniotic fluid and placenta enable the safe growth of the fetus. The amniotic fluid is going to provide cushioning and protection against bumps, while the placenta is going to provide nutrients and oxygen and take away the waste products like carbon dioxide and urea. So I said amniotic fluid protects the fetus by cushioning it from bumps and shocks, and it maintains the constant temperature. The placenta allows the exchange of substances between the mother's blood and the fetus blood, which is oxygen and nutrients which move in, and carbon dioxide and waste that move out. The placenta also produces hormones to support pregnancy, hormones like progesterone, and it has a barrier function, reducing the risk of harmful substances that pass to the fetus. Then in C, scientists investigated the effect of tobacco smoking and taking mineral iron supplements on the growth of developing babies during pregnancy. The scientists looked at 4 groups of mothers. There is Mother A, non-smokers, taking mineral iron supplements. Then B, non-smokers, not taking mineral iron supplements. C, were smokers, taking mineral iron supplements. And D, smokers, not taking mineral iron supplements. So they say the graph shows the mean masses of developing babies at 12 weeks of pregnancy, at 24 weeks of pregnancy, and at birth. We see for all groups the mean mass increases, but group D, those who were smokers and did not take any mineral iron supplements, had the lowest mass, because if they are smokers, then their blood, or the blood, is going to carry a lower concentration of oxygen, and if they do not take any mineral iron supplements, meaning the iron, they are lacking the iron, iron, which is going to be helpful in making hemoglobin, which transports more oxygen in the body. So this is affecting the growth of the fetus. So they say calculate the percentage difference at 36 weeks of the mean mass of babies from mothers in group A compared to the mean mass of babies from mothers in group D. At 36 weeks, group D had 2,200, and here we have 2,800. So the difference is 2,800 minus 2,200, giving us 600. The percentage difference should be that divided by the 2,200 times 100, giving us about 27% to the nearest whole number. Then comment on the effect of smoking and the effect of taking mineral iron supplements on the growth of babies. Use the graph and your own knowledge to help you answer. When people smoke, the carbon monoxide in the smoke binds strongly to hemoglobin, and so less oxyhemoglobin is going to be made, so the fetus receives less oxygen and less aerobic respiration, less metabolic reactions, and less making of proteins, or less increase in mass. But when people take iron supplements, some of these minerals are going to aid in the formation of more hemoglobin, so there is a chance that more oxygen could be taken up, and that little bit increases in the body weight, as we saw, the body mass, as we saw. So I say the graph shows that group A, the non-smokers with supplements, had the heaviest babies, indicating the most favorable growth conditions. Smoking reduces oxygen supply to the fetus by reducing oxygen carrying capacity of blood due to the carbon monoxide binding to hemoglobin. Aerobic respiration decreases, limiting energy for growth, hence lower birth weights. Mineral iron supplements support healthy development, such as for bones and blood. Babies of mothers who took supplements grew better than those who didn't, even among smokers. In question 5, the sequence of bases on one strand of a section of DNA is that. Which of these is the complementary sequence of DNA on the other strand of the double helix? We know thymine always pairs with adenine, adenine with thymine, cytosine, guanine, cytosine, guanine, guanine, cytosine, thymine, adenine, then adenine, thymine, thymine, then we see thymine, adenine, thymine, adenine, adenine, thymine, thymine, adenine. So it's going to be A. Then they say which of these is the correct summary of the process occurring during protein synthesis. During protein synthesis, DNA is going to be transcribed into messenger RNA, and then the messenger RNA is going to go through translation to form the protein. So the correct sequence is A. The table shows different messenger RNA codons for some amino acids. So here we have in the table, they say UUU, AUG, and UGU is one combination of codons that codes for the sequence of amino acids WXY. Give the number of other combinations that code for the same amino acid sequence. The first was this, that, and that. The second one is that, and that. The third one is going to be that, and then that. meaning we have 4 in total. 1 is already given and the other 3. And then they explain why most genetic mutations have no effect on the phenotype of an organism. Use the information in the table and your own knowledge in your answer. This is because some mutations occur in recessive alleles that may not be expressed if there is a dominant allele. Some mutations occur, but they lead to exactly the same sequence of amino acids. For example, if a mutation occurred converting this U into a C, we will still code for the same amino acid W, and so that will have no effect. So I said many amino acids are coded for by more than one codon. A mutation may change the base sequence but still code for the same amino acid. For example, amino acid W is coded for by UUU and UUC, so the protein is unchanged. Some mutations occur in the non-coding part of DNA, and some mutations occur in recessive alleles which are not expressed. Then state one way that the incidence of mutations can be increased: exposure to ionizing radiation like X-rays, UV, or mutagens like those in tobacco smoke and benzene. In question 6, the photograph shows a fish called tilapia. This type of fish is often grown in fish farms. So that's the tilapia. They say tilapia are a good source of nutrients for humans. The table shows the percentage of the daily requirements for humans of 3 nutrients provided by a portion of tilapia. So lipids, we see the percentage of daily requirements provided by this is 16, protein 38, and carbohydrates 16. They say use the table to determine the ratio of lipid to protein to carbohydrate. Give your answer in 1 to n to 1. Of course, we'll just do 16 to 38 to 16. Divide by 16, you get that. Divide by 16, we get that. And divide by 16, we get that. So this should be 2.4. Then describe how to test a sample of tilapia to show presence of a lipid. Here we could crush the tilapia and then we add ethanol. Adding ethanol allows any lipid present to interact with the ethanol. We shake well and then we pour into water. This could be cold water. If a white cloudy emulsion is formed, it indicates presence of lipids. The next, tilapia has high levels of protein. State one functional protein in the human body. Proteins are used for growth and repair, making more tissues, for making enzymes, hormones, antibodies, and so many more. Then in part B, scientists have produced genetically modified tilapia that grow faster. Scientists investigate the growth of these GM tilapia compared with non-genetically modified tilapia. This is the scientist method. Set up 2 equally sized tanks containing water. Put non-GM tilapia into one tank. Put an equal mass of GM tilapia into the second tank. Feed the fish in each tank the same mass of protein pellets. The mass of each type of fish was measured at the start of the investigation and after 7 months. A measure called the feed conversion index was also calculated for each type of fish. The table shows the scientists' results, which are these ones here. So they say the mean rate of increase in mass of non-GM tilapia during the 7 months is 2 to 6 grams per month. Calculate the mean rate of increase in the mass in gram per month for the GM tilapia. Give your answer to 3 significant figures. So we go to the GM tilapia, we see it changed from that to that. That means the increase is going to be that minus that, giving us 2,500. And the rate is going to be the increase divided by the number of months. This was in 7 months, so I got 357.14. But to 3 significant figures, it's going to be 357. Then the feed conversion index is a measure of the mass of protein pellets used compared to the increase in mass of tilapia. It is calculated using this formula here. They say use the information in the table to calculate the mass of protein pellet given to the non-GM tilapia. So for the non-GM, from the table, we see it changed from that to that. So the increase is going to be to 830 minus 1 to 50, giving us that. And from the table, of course, this value here is 1.9, which is equal to the mass of protein divided by the increase in mass. So mass of protein is going to be that times that, giving us that. And so that's the answer. Then they suggest why GM tilapia have a lower feed conversion index than the non-GM tilapia. This could be that they convert food faster into mass, or they have more efficient ways of digesting, or they require less feed. So I say GM tilapia grow faster and convert food into body mass more efficiently. They have lower metabolic rates. They digest food more efficiently, so they require less food for the same amount of weight. And then they say both groups of fish were fed the same mass of pellet and placed into the same size tanks of water. Give one other abiotic factor that the scientists should keep constant. It should be temperature, pH, oxygen levels, light intensity, and more. Then the scientists concluded that farming GM tilapia is better for the environment than farming non-GM tilapia. This is because GM tilapia would result in less nitrate in the water and cause less spread of disease. Evaluate this conclusion and use information in the table and your own knowledge to support your answer. If there is less nitrate, then the chance of eutrophication is going to be decreased because less nitrates, less decomposition, and more oxygen is going to be retained within the water, less chance of eutrophication. So I say GM tilapia grow faster, 357 gram per month in comparison to 2 to 6 gram per month. So less time and feed needed per kilogram of fish. Lower feed conversion index means less food or less feed is used, so fewer resources are required. Less feed means less fecal waste. For example, nitrate in water reducing pollution. There will be less decomposition, more oxygen in the water, less eutrophication. There will be less time in the tanks, and this could mean lower risk of disease spread. However, possible risks of GM organisms include escape into the wild populations, effects on ecosystem, and ethical concerns. So this brings us to the end of question 6 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.